which is from Fernando Vieira, who, as we all know, is the CEO of ALS TDI, hardly needs any introduction after this two-day symposium. He's going to discuss frontiers in ALS therapy development. Fernando.
Thank you, Bob, and thank you all for your attention. I hope you're not all tired of hearing from me yet, but you've got one more round, I suppose. Let me put this into slide mode. So it really has been an honor and a privilege to participate in the ALS One Research Symposium this year. And specifically, I'm excited to present during this session, which had been slated as the ALS community update and talking about new wave of therapeutics and beyond. As Dr. Merit Cudkowicz referenced yesterday, there are probably about 250 preclinical or clinical programs in the pipeline right now for ALS. And so I'm going to try to talk to you about those new frontiers in ALS therapy development, and which is essentially what we've been doing for the last couple of days, right? So all of these presentations have been that. So I'm going to try to take some of what you might have heard today and yesterday and put it into broader context, kind of step back. And then I'll actually take a step further back outside of the content of our symposium today, and look at other drugs that may be in the pipeline across various stages of development. And we're definitely going to talk a little bit about the work that's being done at ALS TDI, ALS Therapy Development Institute. After all, it is in our name. And we are proud that we've developed something of a discovery engine to do this exact job. But I'm not going to go too in depth around our own programs. I hope to be able to do that in a future forum. So I'll start by just saying, like, what we come away from the past couple of days, like we have, is that it hits home that really we're dealing with ALS as an incredibly complex syndrome. It's a disease certainly defined by the fact that it affects both your upper and lower motor neurons, right? So the compartment in your motor cortex in your brain and lower motor neurons in your brainstem and spinal cord. But it doesn't necessarily do it the same way in everybody. It is somewhat of a heterogeneous disorder, particularly at disease onset. You might have one person whose disease symptoms initiate with almost exclusively upper motor neuron symptoms and in the limbs, and that might be initially diagnosed as primary lateral sclerosis and ultimately get diagnosed as a full-blown ALS. You might have a different person who has bulbar onset, so it affects their mouth, neck, throat, ability to speak and swallow, and that might be bulbar ALS. And then we have the more typical presentation, the spinal onset with both upper and lower motor neurons. So it is certainly a complex disorder, and we don't really have a clear understanding of why there is this variability from one person to the next. But we don't have a dearth of hypotheses or plausible explanations for this type of variability. And all we have to do initially is focus in on genetic variants or familial or directly inherited versions of ALS. And this is an outdated table, which I'm sure some might argue could have actually 50 genes at this point that have been implicated in ALS. But despite that, even looking at this more truncated, outdated version, you end up seeing a lot of different genes with a lot of different functions that are implicated. And that suggests that we're dealing with a disease where you could have multiple different initial dominoes that might tip that trigger that cascade that gets you to ALS. And whether there's full convergence or not in what that disease looks like, which was discussed earlier today in one of our earlier presentations, is unclear. But they do certainly all end up in a situation where you have upper and lower motor neuron loss and muscle atrophy. If you take a step back further and look at the molecular and biological processes that have been implicated in ALS, specifically from cell-based models, animal models, and in people with ALS, post-mortem tissue and beyond, we see that really massive amounts of human pathophysiology have been implicated, but we can go through some of them here. And these are themes that you've heard about over the past couple of days. We see nucleocytoplasmic transport defects. We see RNA binding protein and RNA metabolism dysregulation. We see protein aggregation, which we've talked a lot about. We see DNA damage and DNA damage response, DNA repair. We see mitochondrial dysfunction and oxidative stress, oligodendrocytes, which were just mentioned in the last talk, microglial dysfunction. So the immune system is involved in this disease as well. Cytoskeletal disturbances have been implicated. So sort of the infrastructure of the cell. There are genes that have been mutated that play a role in vesicle transport. So consider this almost like the UPS trucks of the cell. So messaging from one cell to another, proteins and RNAs. And then the old sort of standby that we've talked about a lot over the past few years, excitotoxicity. So there are so many different processes engaged in ALS. And really where this leaves us is in a situation where we know we have a devastating condition, right? And we know that it has an unexplained variable clinical presentation. We know that we're dealing with an incredibly complex biology. And this actually gives us a lot of different opportunity because this presents us with many plausible therapeutic targets. And for those of you who might not know, when we talk about a therapeutic target, we're talking about something biological that we want to change, that we want to influence. We want to inhibit, make go up, make it go down in some way so that you can have a therapeutic benefit. And then, and where that leaves us is an obvious need for multiple treatments. And we may need these multiple treatments for a few different reasons. We might want treatments that are targeted at specific patient populations that will only work for those patient populations. But we might also want multiple treatments so that we can stack effects on top of one another, so that we can have a bigger benefit for more people. And so that's our objective. That's our collective objective, I think, of everybody here, is to get to this point. And so taking a step back on what we've heard today, we've heard from multiple different companies just here at the ALS One Research Symposium with many different therapeutic approaches. And this highlights, at least for me, really that we have so many more options in our therapeutic toolbox than we had many, many years ago. And so I'll try to put these molecules that you heard about, or approaches that you heard about today, into some bins that can give you a little bit of context about what we're looking at. And I apologize in advance right now. I presumed that the RAC1 knockdown was going to be an oligonucleotide, Neil. I should have presumed that it was going to end up being a biological. But I'll talk about this now. So what do I mean when I say tools in the toolbox? Well, about 20 years ago, the main tools in the toolbox for addressing a disease like ALS were really limited to small molecules and maybe protein biologicals, right? We didn't really have cellular therapeutics as an option, or oligonucleotides, or things like nanocrystals, or another sort of tool, which I'll mention in the next slide, which are gene therapies. Those weren't there. All we had were these options. And 40 years ago, really, all we had were small molecules. So when we talk about the differences between now and when Lou Gehrig was diagnosed with ALS and the potential for progress now versus then, really, we have so much more potential now because of the tools that we have available. So I want to bring to your attention a whole collection of other therapeutics that weren't mentioned yet today from various different groups that really are in the pipeline and illustrate all of the new tools that we have at our disposal. So I'll start here in the top right corner and mention cellular therapeutics. Obviously, we heard about the brainstorm neurone trial today, but we see multiple cellular therapeutic approaches in clinical development right now. Koya Therapeutics and Rappa Therapeutics are both developing Treg-based or regulatory T-cell-based therapeutics for ALS. These are largely building upon findings that regulatory T-cells may be therapeutic and beneficial in people with ALS. This was work really pioneered by Dr. Stan Appel over the past 20 to 30 years, and it's exciting to see these move forward. We see another autologous stem cell program in an academic collaboration between Mayo Clinic and Regenerative Medicine of Minnesota. Moving on to gene therapies, which are new and weren't really available to the community years ago. Apic Bio is currently leading the way here with a gene therapy approach targeting SOD1, superoxide dismutase, in familial SOD1 patients, trying to reduce expression of that. Again, now building on a lot of work done by Dr. Robert Brown, our moderator for this session, and multiple other programs in preclinical development, one of which from the Institute of Myology in France, ALS Therapy Development Institute worked closely with them to validate a viral gene therapy in preclinical SOD1 mouse model of ALS that was designed to knock down SOD1. And it really did a fantastic job extending the life of SOD1 mice better than anything that we'd ever seen before. And that ended up resulting in the Prize for Life treatment prize for that program. And we hope to see that move forward. When we shift over into the oligonucleotide space, again, a newer technology in therapeutic development being brought to bear in ALS, major investment by Biogen and Ionis in this space currently, both in collaboration and independently. A lot of us know about the tofersen program, knocking down again superoxide dismutase using an antisense oligonucleotide. Biogen also has in its pipeline, in partnership with Ionis, an antisense or an ASO targeting C9orf72 in patients with that mutation, and also ataxin-2. And Ionis is independently moving forward in a trial with JC Fusen targeting people with FUS mutations, familial ALS. And then we see Wave Therapeutics coming in with a different type of ASO chemistry, again, targeting C9orf72 mutation in ALS and frontotemporal dementia. We take a moment and shift up to protein biologicals. So what do I mean by peptides or biologicals? So these are drugs that are essentially strings of amino acids or entire proteins that can be developed and turned into a drug. They usually need to be injectable. And we see a number of these molecules in development today, targeting complement pathway. These typically are taking advantage of the immune system or targeting the immune system. These two programs here in purple, AT1501 is one that I'm particularly proud of. ALS Therapy Development Institute invented this drug. It is a monoclonal antibody. So it's an antibody targeting a protein called CD40 ligand, which plays an important role in innate immunity and adaptive immunity. And we tested a monoclonal antibody like that in SOD1 mice. And at the time, it was the largest therapeutic benefit or life extension that we'd ever seen in that model. And it remains one of our most effective molecules to date. We're very happy and proud to have licensed that to Eledon Pharmaceuticals, who are doing a fantastic job and are running that in a phase 2 clinical trial today. I also want to mention AP101. This is a program that was born out of a company called Neurimmune in Switzerland and has now spun forward into ALS Pharma. This is another monoclonal antibody, which is targeting misfolded SOD1, but it's not limited to being used in people with familial ALS with SOD1 mutations. ALS Pharma is looking at sporadic ALS as well and testing this. It is now, we're actually really excited to see that it was announced that it has moved into a phase 2 clinical trial for ALS that's enrolling now. And then shifting over to sort of the still largest collection of molecules, the small molecule drugs. And small molecule drugs do remain a large collection because there's a well understood pathway for drug development with small molecule drugs. These are the drugs that are most likely to be found in your medicine cabinet or still be prescribed to you today for non-ALS conditions. And there's still tremendous opportunity with small molecule drugs, especially going after newer targets that have been ignored in the past in the ALS space. One of those, again, pointing to Biogen, we mentioned nuclear pore or nuclear cytoplasmic transport. The molecule 100 from Biogen, it targets that. MT Pharma is looking at an oral formulation of edaravone to provide another option for that drug, edaravone, Radicava, that is not intravenous. A small company has a preclinical small molecule program, Econiso, looking at HDAC6 inhibitors with the idea that these would ultimately target microtubules, so sort of the infrastructure of a cell and improve that. We can look at copper ATSM, a drug in clinical trial in Australia, which multiple groups, including our own at ALS TDI, have demonstrated are effective preclinically, but we're really excited to see how that might play out clinically in the current trials. I'm not going to speak much about verdiperstat and zilucoplan because I assume Dr. Paganoni will be touching on those going forward. But we also see Novartis and Arete discoveries, both with molecules targeting the immune system, and follow on the conversation yesterday by Drs. Dubnow and Dr. Nath, looking at antiretrovirals in a trial in people with ALS, a collection of antiretrovirals being looked at in trial now. So we don't have a dearth of programs in development and potential for benefit in people with ALS. It's actually an incredibly exciting time. I do want to bring your attention to a couple more programs out of ALS TDI that I think have major potential to get into the clinic soon. We have a collection of redox modulators, which in our human-induced pluripotent stem cell models rescue mitochondrial function and are demonstrating high effectiveness and efficacy in SOD1 mouse models. So it's making them live longer. And we are working hard to identify a lead molecule that we can move forward into the clinic soon. And then a second program that's not quite as far along, that's still in the drug discovery stage, but it's focused on C9orf72 biology. And this is a new space. We're looking at PRMT inhibitors, so protein arginine methyltransferases. And these are enzymes that modify arginines, which are a specific amino acid that happen to be enriched in dipeptide repeat proteins that are toxic in C9orf72 ALS. And these drugs are effectively completely rescue a cell-based model of C9 dipeptide repeat toxicity. So we're very excited to try to discover new chemical entities that would have CNS distribution and would be effective in animal models to move forward with that there, again, with a focus on C9orf72. So you can sort of reorganize all of these into different categories of therapeutic approaches. I'm not going to belabor this because I just went through all of this, but you can see that there are so many different approaches that are being brought to bear now. It's an incredibly exciting time. I'll summarize in a series of slides going forward that emerging themes in ALS therapy development are that we have a really diverse technological toolbox that's currently filling the ALS drug pipeline. We've got small molecule drugs, biologicals, oligonucleotides, gene therapies, and cellular therapies. The knowledge of ALS genetics is influencing therapy development and patient stratification. So there are multiple treatments that are focused on gene suppression, so suppressing mutant proteins that are likely having a gain of function and are toxic. ASOs and viral gene therapies are central to this emerging trend. And it's worth noting that some non-oligonucleotide and non-gene therapies are using genetics as patient enrollment criteria. So they're using information about a person's genetics to select patients that might be most likely to respond to a small molecule drug, for example. In terms of cellular therapeutics, this is definitely a growing area of clinical development, and there is an increased focus on immune modulation using this technology. These cells might have the ability to sort of non-linearly coax the entire system into a more healthy state, and it'll be very interesting to see how these therapeutic approaches play out over the next few months and years. As far as protein biologicals in development, their monoclonal antibody therapies are making up the majority of the biologicals in development for ALS. There are a few different approaches that are targeting the complement system. So it'll be, again, very interesting to see as these therapeutics move forward. We'll get a clear sense of the role that complement might play in ALS and its ability to be targeted for a benefit. And really, nearly all of these biologicals in development for ALS use or take advantage of the immune system. And then a micro trend that I want to point out, and we heard from two folks today, Amylyx and Neurosense, sort of bundled therapeutics. So combination of two drugs bundled together into a single therapeutic approach to see if you can have enhanced efficacy. This might be a growing trend. We, again, assume that we're going to need multiple therapeutics to make a difference here. We don't have to start every trial with drugs stacked on top of each other, but that is definitely the direction we can anticipate going. So it'll be interesting to see how that plays out as well. And the majority of the drugs that are in development remain small molecule drugs with hoped for benefits for all people with ALS. So despite the fact that there are these more stratified and targeted approaches with all of these newer technologies, the still biggest feeder to the pipeline is our therapeutics operating under the assumption that ALS will have common pathways of biology that are engaged in their disease that will be treatable, even if incompletely, by the same therapies. These approaches have the benefit of clear therapeutic development paths, and so they'll remain common, and that's a good thing. I just want to point out that ALS TDI ourselves, we certainly keep an eye on the entire landscape, and we don't operate in a vacuum, and we have many, many partners, many of you who are likely on the call today. and looking forward to building stronger relationships going forward and continue to work together to end ALS. And I didn't go into detail about ALS TDI's research programs today, but I did touch on them. And I want to acknowledge all of the people past and present who have worked very hard to develop all of those programs and to develop ALS TDI into what it is today, an institute that I'm incredibly proud of and that I look forward to stewarding to contribute to the end of ALS. And with that, I will open it for any questions.
Thank you. Thank you really very much, Fernando. That's marvelously both backward and forward looking and much appreciated. I'm going to start with a question or two first, and then obviously we'd like to throw this open to anyone. You picked up on what I was going to say is a big issue here, and that is combinatorial therapies. So you might want to comment, maybe Dr. Paganoni will as well, but the old rule of thumb was they were always going to be difficult because one had theoretically to try one drug at a time, then the other, then put them together. And the concept was that strategically this would represent great increases in the size and cost, of course, of executing a study. But we've seen these, and I think a little more so far in the AMYLIC study, in fact, really something quite unexpected, and that is using one set, and this came up as part of the discussion, one set combination of two drugs at a given dose, carefully worked out ahead of time, tried without any of the other single drug by drug comparative studies. So can you comment on the challenges in doing the combinatorial studies? Because it's quite stunning that those are remarkably absent from everything except for the two cases you've illustrated.
I can speak to it from the perspective of trying to do it preclinically. And it ends up being an exponential effort, right? Trying to sort that out. When we look at molecules preclinically in cell-based models and then ultimately in animal models, you end up having to do careful dose range studies independently, looking to see if you can hone in on the most efficacious concentrations and doses in each of those models. And then you try to, at least the way we think about it, you try to set a ratio and then dose range the ratio from there. And suddenly you go from having something that is practical, that you can actually execute in your lab and won't consume all of your resource, to something that's a massive effort that consumes just about everything that you do. And if you're maybe if you're a company with a single focus, and that focus is those two drugs, you can invest those resources to do that preclinical work up front that can guide those clinical studies. But it really is incredibly challenging. It's a big issue to confront. And I don't know how the clinical pipeline could will end up being able to accommodate those types of studies in the size that they might require. But we do have a couple cases of potential that are in the pipeline right now. So we'll try to learn lessons from those.
Yeah. Thank you. A question that's asked is, as you go through the pre-clinical assessment of a compound, what are the major hurdles that you encounter? And I want to maybe broaden that a little bit and ask, as you think about the preclinical pathway, let us say before about a year ago versus now, do you see fundamental changes in the way the FDA will view the preclinical hurdles to make things easier? For example, do you think it's possible that we will be able to get away from the insistence on not one, but sometimes two large animal studies prior to going into a human trial? And what do you think the future is for that?
Okay, so I'll try first by answering your first question, which is the biggest hurdle is finding something that works. So we're here today and people are presenting their best, right? It's where we're all presenting the information of the things that probably worked. And I can speak for in our lab, When we test things in our various models, cell-based or animal models, I would say 95% of what goes into an experiment doesn't do what we hoped it would. And that's okay, because you learn something and you move on. So that's the biggest hurdle. Then the follow-on hurdles are, if it works in that model system, does it work in the more complex model system? Right? And oftentimes you have attrition at that point. And then sometimes if it works in that more complex model system, it also does something you didn't want it to do, like liver toxicity or something like that. So there are many, many hurdles that go from an idea that's a good one, all the way to a drug that you're trying to get into people. And then to have it work in people in that even more complex system is a bigger challenge. Now, your question about sort of the systems in place and regulatory hurdles, I can speak less to that, but I have the impression, looking at the overall ALS landscape, that there is more flexibility, and particularly around the idea of demonstrating that you have target engagement and being able to make a case that your target is a valid one in the ALS space, and particularly in a specific patient population. I think there's more flexibility there, and I think that's a good thing.
Thank you. Just a comment from Dr. Peter Werner vis-a-vis this question of combinatorial studies. What he says is, and he's completely correct, that it would be beneficial if we had access to a representative of the FDA. The old rule is that he writes, I had to do one compound alone, then two alone, and then the combination, and then all versus placebo is mainly a rule that's based on the regulatory agency's stance and not necessarily on optimal pursuit of the biology. And I think that's a very valid point. I want to bring up another issue, which actually has come up in several of the chat comments, and that is, how do we go about sharing the kind of richness of information about the pipeline that we all work with our own separate private spreadsheets, with the general patient community? Is there and I know TDI probably does a lot of that, but we can do better. Can you comment on providing access to this information, Fernando?
Well, I think forums like these are a good one, right? Where we have this opportunity to share these types of information. I think there are multiple nonprofit organizations that are trying really hard to do this. I think the ALS Signal dashboard does a pretty good job demonstrating what's in the clinical trial space, and ALS TDI also has a list of what's in the clinical trial space. The preclinical space is harder to keep tabs on because a lot of things are happening. There are stealth operations that are deliberately not out there. And then there's almost who's spending all of their time making sure they got everything, right? It's so much of it is what information crosses your path. You can be actively looking, but a lot of the information passes in front of you. So curating and sharing all of that is a Herculean effort. I do think more can be done, and I think we and others are undertaking to do that, but to try to get databases available that keep track of this.
Thank you. Yeah, it's a huge issue. I mean, I find it something that I struggle with virtually every week, trying to update my own little spreadsheets, but I can only begin to imagine the frustration in, as it were, the consumer community, patients who really want to know that information. So your points are well taken. Well, that's wonderful. Thank you again, Fernando. Let's turn now to our final presentation, which is